In this video we will discuss about a new area, that is bacteria can be used as a biomarker for early tumor diagnosis. So this is you can consider it as a new field and at this point of time. So we will discuss how this bacteria, why people think that bacteria can be used as a biomarker for early diagnosis. So recently what people have found out is that there is a microbiota of the tumors. The tumor also has some kind of bacteria, and how these bacteria approaches to the tumor cells. There are different ways or different possibilities that are shown in this figure. First one is through the this decreased or damaged mucosal barriers. So the tumors usually cause the change in mutations, and that might leads to the damaged mucosal barriers. And the bacteria, the first line of defense for the bacteria to infect the patient or human beings is these mucosal barriers. So those opportunistic bacteria will then pass through the mucosal barriers and reach to the tumor cells. This is first line of defense. And in order to support this point, people have seen there is a upregulation of ST6 galnac-1, and that will cause to the disruptions in this mucosal barriers. The next way is through this oral cavity or intestine, and after that this the bacteria has some kind of receptors that has more specificity towards specific carbohydrate structures that are expressed on the tumor cells. For example, this F nucleotum that we have discussed in one of our previous video, F nucleotum bacteria has this receptor FAP2, which has a high specificity towards gal to galnac carbohydrate structures, and these gal to galnac carbohydrate structures are overexpressed in the colorectal cancers. So this F nucleotum can enter through the oral cavity and can reach to the tumor sites, or if the patient has colorectal cancers, then those F nucleotum bacteria can accumulate at the colorectal cancers, and this is the reasons the colorectal cancers. This could be the reason the colorectal cancers has high microbiota in term, and which is mainly dominated by F nucleotum. The other things is through the gut organ axis, and in normal cases what happens, there is a continuous transfer of microorganisms from gut to the different organs, and there is a recycling of microorganisms from different organs to the gut, and this could even cause, and these disruptions in this can even be because of the change in the uh, change in the change in the functioning of any organs because of the generation of tumors or because of the mutations that occurs within the organs. And for examples, in the case of pancreatic cancer cells, people have found out there is upregulations of one kind of bacteria, mucosus, and not able to recall its complete name, but that bacteria is found to be upregulated in the pancreatic cancer cells. And however, in the normal pancreas, they have not seen the upregulations of that mucosus bacteria. So this could be because of the mutations that occurs within the tumors that attracts or prevents the recycling of the microorganism, this microbiota or the bacteria back to the gut. The last way by which these tumors can spread is through the tumor microenvironment. As tumor microenvironment has a different vascular system, and it is also rich in nutrients and has hypoxia conditions, which is kind of favorable to the bacteria. So they are more prone to remain in the tumor microenvironment than to their compared to the other positions. And this is one of the reasons by which this microbiota can be expanded or can result in change in the tumor, the bacterial compositions near the tumors. So these are some of the 4 ways by which the microbiota of the tumors can be disrupted. However, this is at the initial stage. This idea is at the initial stage, but it can be act as a biomarkers or one of the possible candidates for the biomarkers to detect tumors at the early stage. So that's all for this video. Thank you for your time. If you need to understand more about this in detail, you can go through the reference that has been provided in the description.